So, let us move towards the last phase, which is known as termination. Termination is different in both prokaryotic and eukaryotic cells. Prokaryotic cells have two types of termination processes. One is row-dependent, and another one is row-independent. In row-dependent termination, row protein will kick the RNA polymerase out, and the RNA will be stopped synthesizing. Then, in row-independent termination, if a particular sequence of nucleotide occurs, the nucleotide gets attracted towards each other and form a hairpin loop. After hairpin loop, RNA polymerase detaches from the DNA template, and hence the process is terminated. Now in eukaryotic cells, termination occurs through signals. What happens is that when a DNA template is forming, there is a specific sequence that it reaches. That sequence is A, U, A. This sequence is known as a polyadenylation signal. This polyadenylation signal activates certain proteins that stop the process of transcription. The remaining formed RNA detaches from the DNA template. So, this concludes our video on cell transcription. The next video is going to be about cell translation. So stay tuned.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.